It's Max Kramer, faculty for Biology 1961, Foundations of Biology, and this is part two of the Exercise 9 video background series. So you'll remember in part one, we talked about alpha diversity, and that is the diversity within one sample or one community. So we talked about some examples of that. We talked about, say, the number of tree types in a defined plot of forest, say 1 mile by 1 mile. We also talked about, most importantly for this research project, the number of different OTUs or species in a stool sample. So that's a measure of diversity within one microbiome. So for this video, I'll be covering beta diversity, which I introduced a very little bit about, but I'll go in a lot more detail here. So that's the change in diversity between two samples or communities, or along a gradient. So you're comparing things to each other. So you need at least two different things to compare to each other. So we might look at something like the composition of a forest along the slope of a mountain, the lower elevation versus higher elevation parts of that community. Or we could look at, again, using a microbiome, gut microbiome as an example, we could look at the composition and the community members in a stool sample versus the microbiome of the skin. So if you took a sample of the microbiota from skin and compared that to a stool sample. So the big three take-home ideas, which the numbering is a little messed up, but it should be 1, 2, 3, not 1, 1, 1. The first take-home idea is that beta diversity compares diversity found in two different samples. The second idea is that diversity metrics are ways to compare different points of emphasis that represent the diversity between two samples. This could be the number of species present, the number of different species or taxa present, the phylogenetic relatedness of taxa present, or incorporating both the taxa abundance and the phylogenetic relatedness. So this is pretty similar to the alpha diversity metrics that we talked about last time. There's a parallel to beta diversity metrics. And then lastly, for beta diversity comparisons among many different samples, we can represent those visually using PCOA and clustering plots, which is a way to visually see how different samples are from each other. All right, so like I've kind of been hammering home over and over, alpha diversity is the diversity within a population. So the alpha diversity of these two plates, plate A and plate B, are defined independent of each other, whereas beta diversity is comparing these two plates. So a few different comparison points between these two ideas of alpha and beta diversity. For alpha diversity, we can look at how diverse one sample is. We could look at the number of taxa present. We could estimate the number of taxa in each sample. You might remember rarefaction is helping to make those estimates the same for different samples or the same sample size. We could take the average number of taxa found at different body sites. We could do something like, hey, we found 20 different taxa in the stool sample, but only 10 different taxa in the skin sample. Whereas with beta diversity, again, you're comparing how different those samples are from one another. So you could look at the overlap between two samples, how many shared taxa are there between those two samples. You could take the average of two different groups. If you have lots of samples, like you have 10 gut microbiome samples and 10 skin microbiome samples, you could take the average difference between samples. But you're making those comparisons. So here's a case where we have alpha diversity. Oops, just going to hide this. We have alpha diversity of 4 different bacterial plates with the little circles on the plates representing different bacteria types and the colors representing different taxa or different species of bacteria. So in each of these plates, we have lots of different kinds of bacteria. Lots of different colors, there's kind of a whole rainbow. So there's high alpha diversity within each of these plates individually on their own. But if we switch to beta diversity and we make comparisons, we might compare these first two sets of plates. Those have lots of different colors in them, but the distribution of colors is pretty similar between the two plates. So we'd say this has low beta diversity between these two samples. Whereas these two plates, oops, skipped ahead, these two plates have lots of different colors on each plate, and those colors are different between the two plates. So this sample also has a high beta diversity between these two plates. So how do we actually calculate beta diversity in a quantitative way? We're always making a comparison between two samples, and there are lots of different quantitative mathematical metrics we can use to calculate beta diversity between two samples. We're going to focus on two particular ones called Bray-Curtis and Unifrac. And so what you end up doing is you make pairwise measurements for all the possible combination of sample pairs, and you put those measurements into one new matrix or table. A matrix is, in this case, just another name for a table. So you have all your samples on one in your rows. You have, again, the same samples in your columns. And because you're comparing them, this matrix or this table is actually symmetrical. So if you go along the diagonal, you're comparing sample 1 to itself. So it has a high 1, in this case, is a low beta diversity. And then sample 2 compared to sample 1 is 0.4, which is the same as sample 1 compared to sample 2, 0.4. So these tables end up being symmetrical. So how do we actually calculate these two metrics? Let's start with Bray-Curtis. Bray-Curtis measures the similarity between samples, and it ranges from 0 to 1. So like I was saying a moment ago, 1 means two samples are very, very similar to each other, or completely the same, which makes sense. You compare a sample to itself, it should be completely the same. So how are we doing this calculation? It looks a little intimidating, perhaps, this mathematical equation, but let's break it down piece by piece. So on top, we have xi and xj. So these are just the abundance of a species within sample I and sample j, two different samples. So first you find the difference between those samples, and then you add those two samples together. So we'll go through an actual example with numbers. But you sum up or you add up all the differences, and you sum up or you add up all the totals, and then you turn that into a fraction. And then you do 1 minus to get the similarity. So what does that look like in practice? Here's a table, an OTU table. Hopefully you're starting to get a little bit familiar with this format. Here are all the samples in the rows, sample 1, sample 2, sample 3, and here are 5 different OTUs that were found within that sample across the columns. OTU 1, 2, 3, 4, and 5. So let's start off with comparing sample 1 and sample 2 to each other. So sample 1 compared to sample 2, we'll start with OTU number 1. First, like I said, we subtracted, so we've got 10 minus 46, and then we're taking the absolute value so that we always get a positive number for the difference. And then we're adding them together, so 10 plus 46, and turning that into a fraction. So in this case, we would have 36 out of 56. It gives you a numerical value. So great, we compared sample 1 and sample 2 for OTU number 1. Now we just need to do that for all the other 4 samples in this particular table. So we've got 10 and 1 for OTU number 2, 10 and 1 for OTU number 3, and so on. This isn't a very creative example data set. It's 10 and 1 for all the rest of the OTUs. So then we add those all up, get a fraction, and subtract that from 1. So if you did all that math, calculated everything out, maybe wrote a little script in R to do that, you would get a quantitative value of 0.28. So that's the Bray-Curtis value between sample 1 and sample 2. So now we can keep going and compare all of the samples. So now we're going to compare sample 1 and sample 3. Here is for OTU 1, 10 minus 10, 10 and 10, and so on and so on. We get a value of 0.6. And we can keep going, compare sample 2 and sample 3, where you get now a Bray-Curtis value of 0.24. So now we've calculated the Bray-Curtis values for all the possible comparisons between our samples. We didn't do sample 1 versus sample 1, but that turns out to be 1. Sample 1 is the same as sample 1. So now we started off with this OTU table, and we calculated all of these Bray-Curtis beta diversity values, and we can put this now in this diversity matrix, or this distance matrix. We've got sample 1, 2, 3, and sample 1, 2, 3, and here are the values that we calculated. 0, 1, 1, 0.28, 0.6, and then the same thing along the horizontal. So any comparison you can go through and look up the comparison between the two samples. So based on this data, if we just look at row number 1, here's my little mouse, sample 1 compared to sample 1, very similar. Sample 1 compared to sample 2, 0.28. Less similar. Sample 1 compared to sample 3, 0.6. So sample 1 is more similar to sample 3 than it is to sample 2. And we can keep doing this for all the samples. And just remember that the more similar, the higher a Bray-Curtis similarity measure. So that was using just the abundance of different taxa within the samples. If you remember from alpha diversity, we also used relatedness, in a phylogenetic tree, to help determine how different or how much diversity was within a sample. So we can do a similar thing for beta diversity when we're comparing two samples. And this metric is called unifrac, which is short for unique fraction. And that unifrac, or unique fraction distance, uses a phylogenetic tree, kind of like this one, like we talked about for alpha diversity, and looks at all of the different tips of that tree, which are the OTUs in the sample, and sees which ones are shared, which branches are shared between two samples, and which ones might be unique to a specific sample. So I'll go through more specific examples in a moment. But the general calculation is just the unique, or the unifrac distance between two samples is the number of unique branches divided by the total number of observed branches, so the unique and shared branches together. So what does that look like? Here we've got two samples, let's say a red sample, excuse me, a red sample and a blue sample. And here are all the OTUs that were observed in either of the samples, all the possible OTUs we could find. And this little symbol over here means that we found OTU number 1 in both the blue and in the red sample. So this has a purple branch because that OTU is shared between both samples. And for this example, let's say we found every OTU in both samples. So we have purple, 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 and then you can go up a level in the branch and you can say, does everything below, is this a unique or is this shared? And this one has blue and red branches below. So this branch will be purple. So there it is written out. We don't have any unique branches in this example, but if we did, they would be red or blue colored branches, and shared branches that have OTUs in both samples show up in purple. All right, so let's look at a second example. In this example, we have our red sample and our blue sample, but this time red sample is totally unique from the blue sample. So all the OTUs in the red sample only show up in the red sample, and all the OTUs in the blue sample only show up in the blue sample. So here, now the branches are red because they're unique to the red, and if you go up a level, this branch is red because everything below it is red. You can keep going all the way up to this very furthest back branch because everything below that branch is red. And then the same is true for blue. So here's a more realistic example. I gave you the two extremes where everything is shared and nothing is shared. So let's look at something that you might actually see in a real example of a microbiome. So in this case, we have our 10 OTUs again, and some of them are unique, like OTU number 1 is unique to red, OTU number 2 is unique to blue, but some of them are shared. OTU number 4, for example, showed up in both the blue and the red samples. So for OTU number 4, this branch is purple. And let's look at the 1 and 2 again. So the first branches, you have just one unique blue or one unique red OTU. But if you go up a level, this branch, if you go down, has blue and red beneath it. So it's actually purple. Same for this one. This one, if you go down, you have some blue and you have some red. So this is purple. So now we can kind of compare the two extreme and the one more realistic example to each other. If you calculate just the number of branches, this one has no unique branches, so its unifrac distance is going to be 0. This tree has all unique branches. It doesn't have any shared branches. So the unique fraction, or the unifrac distance, is 1 for this. So you'd add all these up on the numerator, and then you'd add them all up on the denominator, and you'd get the same amount. Whereas this unifrac distance for this particular, these two samples, you sum up all the unique ones. So there's 1, 2, 3, 4, 5, 6, 7, 8 unique, just red or blue branches. And then you'd sum up all the total number of branches, which I'm guessing is 16. because we calculated a total unifrac distance of 0.5. So this metric is telling you, just based on how related the OTUs are, how similar or different this red and blue sample are. So that doesn't tell you actually something very important about how many of each of the taxa or OTUs you see in your sample. So if we go back, there's no accounting for how many of OTU number 1 were present in the red sample, or how many of OTU 5 or OTU 8. So there's a sort of second version of unifrac called the weighted unifrac distance. So now instead of red and blue, we have a square sample and a circle sample, and we can add in the number of OTUs or taxas we see in each sample. So this one, OTU number 1, had 2 OTUs observed in the blue, or not the blue, the square sample, whereas the very next one, the circle, only had 1 in the circle one. And if we go down here, we have 1 observed in the square group and 2 observed in the circle group. So you take this, our branches, our branch numbers, and we're going to multiply it by the actual abundance of taxa present. So we've gone over 3 different metrics, 1 unique one, Bray-Curtis, and then 2 versions of unifrac, weighted and unweighted version. So you can kind of think about these as what information are these metrics using to decide how diverse or how different samples are. So if we look at unweighted unifrac distance, that uses a phylogeny, but it's not using abundance. If we go down here to the bottom, and remember Bray-Curtis, that didn't use phylogeny, but it did use sample abundance. And then if we look at weighted unifrac, that's kind of incorporates both. So you're using the phylogeny, but you're also incorporating how many of each OTU were observed in each sample. So for all of these diversity metrics, you end up with that diversity matrix, or some people call it a distance matrix, but it's really just a table collecting all those beta diversity numbers. And it's going to compare every sample to every other sample in your comparison. So what do we do with that table? How do we actually learn something from beta diversity or get some information out of beta diversity? It can be kind of confusing to think about because we always are looking at a comparison, and we're looking at many comparisons all at one time if you have lots of samples. So there's 2 ways to visualize this type of data. The first one is a cluster analysis, where you actually take your samples and you make a phylogenetic tree comparing your samples to each other. So the red and yellow sample in this case are very similar to each other, so they're next to each other on this phylogenetic tree, and this blue sample is more different, so it's further away on the phylogenetic tree. So that's a cluster, or a hierarchical clustering. We can also do something called ordination, and we'll talk a lot more about this. But that's a way to graphically represent similarity and dissimilarity between things using points on an axis, so points on a plot. So basically what you need to know is, for this version, points that are closer together are more similar. So here's red and yellow in a hypothetical example are more similar to each other than a blue sample. So these are 2 different ways of kind of looking at the same set of comparisons. So how do we get to those 2 comparisons? We can start off with a phylogenetic tree of OTUs observed in different samples. This kind of looks like the example I was using for phylogenetic distance. I'll just added a yellow sample in now, so now we've got 3 samples instead of 2 samples, and we can compare each sample in a 1 versus 1 comparison. So we can do red versus yellow, red versus blue, and yellow versus blue. And we come up with that distance matrix. And this is a little bit different from the one I'm talking about, but the same concept. And then we can do clustering, or we can do principal component analysis, a version of ordination, based on this distance or diversity matrix. So we're going to start with the ordination version. How do you make these axes and plots? So we're going to start with something called principal component analysis, or PCA. And it's a way of identifying underlying patterns within data that might not be initially apparent. So for our purposes, we'll use the similarity scores from beta diversity, and then we'll do something called compressing the data by reducing the number of dimensions and then plotting that, making a visualization or a graph out of that. You don't need to know the mathematical details of that last point about how or what compressing the data and reducing dimensions is doing. Um, but you can kind of understand that we're simplifying the data and making the patterns underlying the data more apparent. I have this nice little animation here that's a little bit of a visual explanation of what we're doing. So PCA, or Principal Component Analysis, here's my mouse. These blue dots are all some sort of data, and let's just say this was the similarity scores between two samples. And so you have all these comparison samples, and it started off on these two axes. What PCA is doing is mathematically, it's rotating your axes to try to reduce the distance from the points to your axis. So we've got these little red lines connecting every point to the axis, and you can watch it as it rotates. You can kind of see visually that the distances as a whole get smaller or get bigger. And what PCA is going to do is mathematically it's going to stop that rotation when you minimize all the distances. So for this particular animation, you can notice when it gets to be this diagonal, you see very short red line distances. So visually, that's what's happening on a mathematical sort of underlying basis. But we're going to do or use a variation of PCA called PCOA, which stands for Principal Coordinate Analysis. So they're very similar to each other, but PCOA actually is more mathematically suited to handle beta diversity because we can look at similarity or dissimilarity metrics or numbers. And Bray-Curtis sometimes shows up as dissimilarity matrices, and so does weighted unifract. So we need to use this principal coordinate analysis, but it's the same basic underlying idea. It produces a new set of axes that we can make a new graph on to summarize our data sets. But what you need to know is how to interpret a PCOA plot, and it's pretty straightforward. Points on the plot that are closer to one another are more similar to each other than points that are further apart. Simple as that. So let's look at some examples. Here is the result of a PCOA plot. So we've got some comparisons, and now these axes have been relabeled PC1 and PC2, which are kind of mathematical definitions. They don't mean anything necessarily definable in the real world. They're a mathematical definition. So what we can do is we can look at the comparisons between blue points and the comparisons between green points. So in this case, visually, just looking right away, you can see the blue points are closer to each other and the green points are closer to each other. So the blue ones are similar and the green ones are similar, and there's not much similarity across the two different types of points. So how do we actually quantify that? We can calculate the physical distances. So here's the blue points, here's all the possible distances between blue points, and then we just show an average distance within the blue points. We can do the same thing for the green points. And there's an average number or an average distance between all the green points. So within the blue points, it's a pretty small average difference. Within the green points, it's a pretty small average distance. But if we look between the two types, it's actually a pretty large distance between the two types of colors. So here's all the possible comparisons between the groups, and you'd get a larger average distance there. So from that, we can say that these are similar to each other, but they're not too similar to each other, between each other. So we can have all kinds of different patterns when we look at beta diversity on PCOA plots. We can have something where maybe there's a tight cluster of one type and a more spread out cluster of, say, the green ones. So we can measure beta diversity distance again and have a small average distance within the blue, a large distance within the green, so they're less similar to each other, and then kind of in between for the differences between those. So here's an example from a real paper that was published in 2012, and it was, in this experiment, the researchers were looking at different sites of the human microbiome. So they took samples from the gastrointestinal tract, they took samples from the oral microbiome, nasal, the nose, skin microbiome, and the urogenital tract microbiome. And then they did this principal coordinate analysis, and they found that each body site was pretty similar to itself. For the most part. So this is especially true for gastrointestinal samples. They're all very similar to each other. They're not very different. Versus the nose, the nasal passage, those points are more spread out. There's more distance between those points. So the nose samples are less similar to each other. and the nose and skin samples are right next to each other. So the nose and skin samples were actually very similar, but very different from both of those different from the gastrointestinal samples. So we can do another interpretation of some PCOA plots. So this is another example from a real research paper where some researchers were looking at rice roots, the roots on rice plants, and trying to figure out what microbes and bacteria were present based on what kind of farming or what kind of area in the plant they were looking at. So if we look over here, over at this panel D, here's a PCOA plot. And the orange dots are the samples from organic farming, and the brown dots are something called eco farming. And so the bacteria at the root were more similar among the organic and more similar among the eco farming. That type of farming caused a difference in what bacteria were present. And then finally, because this is quantifiable, we can actually quantify those differences. So here's another example of a published figure or published paper figure. So these are all samples from skin, and it's beta diversity, so you're always looking at a comparison. So what's the average distance on the elbow between diseased and unaffected patients? So here's your average distance. What about between unaffected? It's a larger average distance. Or diseased, larger average distance again. So we can, instead of looking at that PCOA plot and sort of estimating clusters, we can actually quantify the length of differences. All right, so that is beta diversity and how we look at beta diversity, how we measure beta diversity. So just to review the big take-home points from today, beta diversity compares the diversity found between two samples. We have beta diversity metrics that we use to compare those samples, and those different metrics have different points of emphasis. So some use the species or taxa abundance, some use the phylogenetic relatedness of the organisms or the taxa within the communities, and some use both of those to create a quantitative or a numerical metric. And then finally, the most common way we look at beta diversity is by using a PCOA plot to visually look at all the different samples and see how similar or different and how samples cluster together. So we use those PCOA plots. Thanks for listening to part 2. Tune in again for part 3 coming soon. Thanks a lot.